Hello everyone. So today we are going to look at a very specific question, that is, what is enzyme specificity? But before we go into that question, so if you have not yet subscribed to my channel, please do that. We will be covering each and every topic of biochemistry in a simplified way and try to answer important questions. So let us continue. What is enzyme specificity? Okay, so enzymes basically are highly specific in their action. Okay, so that means they are their action is predefined. Okay, they have a defined function. So the specificity is mainly of three types. Okay, so the enzymes can be either stereospecific, they can be either reaction specific, or they can be substrate specific. Okay, so let us look at one by one. First, stereospecificity. What does it mean? Now, enzymes act on specific isomeric form. Okay, for example, we have is L and D isomers. Okay, so isomers are basically different spatial arrangements of the atoms. Okay, for example, in L amino acid, the amino group is on the left side, the D amino acid, the amino group is on the right side. Okay, like mirror images these now. So stereospecificity is the different spatial arrangement of atoms, okay, but they have the same molecular formula. Okay, so now enzymes which are stereospecific, for example, L amino acid oxidase will act only on the L isomer, or the D amino acid oxidase will act only on the D isomer. Okay, so this is known as stereospecificity. Okay. Next, coming to reaction specificity. Now enzymes are very specific for the reaction which they catalyze. Okay, for example, transamination reaction is transfer of a amino group. So that is catalyzed by enzymes like alanine aminotransferase or aspartate aminotransferase. So ALT and AST, they catalyze transamination reaction, transfer of amino group, whereas deamination, removal of amino group, is catalyzed by different enzymes. Okay, glutamate dehydrogenase and other enzymes. Okay, so that is deamination, removal of amino group. So based on the reaction, the reaction which they catalyze or which they are specific for, enzymes are again classified as reaction specific. Then the last type of specificity is substrate specificity. Okay, the substrate on which the reaction of the enzymes act. Now it can be an absolute substrate specificity or a relative one. Absolute is, as the name suggests, it is going to act only on that particular substrate and no other substrate. Okay, so it acts only on one substrate. For example, the enzyme glucokinase will act only on glucose and no other substrate. Similarly, urease will act only on urea and no other substrate. So these enzymes are absolute substrate specific enzymes. Next, we have relative substrate specific. So these are the enzymes which can again either act on a specific group or they can act on specific bond. Okay, so specific group is, so enzymes will act on those groups which involve a particular amino acid. Examples, bonds involving lysine and arginine. Okay, so trypsin is the enzyme which will act on bonds which involve lysine or arginine. Okay, so the trypsin enzyme is specific for breaking bonds which involve lysine or arginine. Similarly, we have peptide bond. Okay, bond specificity. So peptide bonds, proteolytic enzymes, they act on peptide bonds. Okay, irrespective what the groups form the peptide bond, they're going to break the peptide bond. Okay, similarly, enzymes which act on glycosidic bonds like glycosidase are specific for glycosidic bond, irrespective of which groups form the glycosidic bond. Okay, so this is an example of relative substrate specificity. Okay, so this is what enzyme specificity is all about and what it means and what are the different types: stereospecificity, reaction specificity, and substrate specificity. So thank you very much. We'll see again.